We may already have the medicines we need for many known diseases. We just don't know which disease they are right for. So a challenge we have now is matching the right therapies to the right patients and disease. I'm Abraham Lin, a biomedical engineer working with patient-derived organoids, living models of patient tumors in the lab. For this hackathon, my goal is to identify and prioritize clinically available drugs that could help rectal cancer patients achieve a response that helps them avoid devastating surgeries. The medicine I identify can be directly tested in the organoid models I work with as a bridge to clinical trials. My hypothesis is that the biology that makes these tumor resistant to treatment and the drug profile of FDA-approved medicines exist in fragmented databases and can point us to the right drugs. And using Claude Science, I came up with two different search methods, both starting from that biology. So most rectal cancer patients have no targeted treatment at all. Everything rides on chemoradiation to shrink the tumor completely, because a complete response is what can let them keep their rectum instead of losing it to surgery. And since the patient population is getting younger at an alarming rate, they desperately need a solution. I started by looking into the specific patient population I was interested in, dividing them into those who respond to therapies and identifying what makes these tumors resistant to them. From there, I developed a plan. Based on what I learned, I searched for drugs using two separate ways. One starting from biology with chemoradiation-resistant transcriptional programs, and one independently from clinical kinase inhibitors comprehensively profiled. Together, they flagged 287 drugs after weaving together 11 public databases from patient data to drug target measurements to safety records. A final filter built from real FDA safety labels narrowed it to two shortlists of 44 drugs ranked safest. But I didn't just run one analysis in Claude. I ran the entire scientific process through it. I used Claude to plan the project from a single clinical question, build the two search engines, pull together all 11 databases, reason through the hard methodology choices, and write the first draft of a manuscript. Then I turned Claude on my own work as a tough peer reviewer to find the real weak spots. I used it to again fix them and rebuild figures, adding a statistical test and tightening every claim. So question, design, data, draft, review, revision, the full loop of science. From here, the real tests will begin, and I can validate these therapies in the lab on cancer models that closely represent human tumors from a biobank of organoids. This will act as a surrogate clinical trial before moving towards patients. This is my way of finding if the drugs we need already exist, identifying candidates with Claude, and testing in the lab. And I hope to turn this method into a pipeline to repurpose drugs for other diseases. Thank you.